Are you here?
Oh,
Okay.
okay.
Is it okay if we start?
Yeah, yes, okay. Thank you. Okay, no problem.
Okay. Thank you everyone that has joined today. Good afternoon. Welcome to the 7th International Lecture Series for Marine Pharmaceutical. Our topic for today is omics technology. I'm very sure that we would gain a lot of useful information from this session. As we know, in the pharmaceutical field, omics has become a technology that is very useful, where it enables us to obtain a global picture of a disease and situation, which then leads us to drug discovery. Before I start, I would like to give a brief introduction of our speaker today, Prof. Kato. Prof. Kato is from Nagoya, Japan. He earned his Ph.D. from the Graduate School of Bioagricultural Science at Nagoya University, where he focused on water tests on plants. He then did research on water channels known as aquaporin at Johns Hopkins University. He was also involved in a research on the discovery of aquaporin with Peter Agre, who has then won a Nobel Prize in chemistry for that research. And Prof. Kato also established a method for selecting and enriching cells by taking advantage of the functions of aquaporins, while at Tokyo Dental College and Keio University, for which Prof. Kato has earned the Best Presentation Award at the Japanese Pharmacological Society annual meeting. Prof. Kato also did research at Peking University and then established Aqua Lab at Keio University. And therefore, without further ado, Prof. Kato, the time is yours.
Oh, thank you so much. The nice introduction is, and also thank you so much invite this lecture. I'm very happy to join in here. So today I talk about for pharmacological method by using the omics. So just a moment. Can I slide? Okay. So today I'm just talking about omics-based pharmaceutical analysis. So previous Angie-san, very nice introduction for me. If you're interested for my detail, please scanning my QR code. I'm also now just joining its Dokuritsu project. It's here. It's for focusing the 2014 year for aging elder person, how to do for healthy. So if you're interested, please scanning here. Also, just one of the showing my memorials pictures. This is Professor Snarabi-san. He is about 25 years ago. I met him and then joining some research and we continue with some good relationship. And I'm very happy to the nice communication that it's at Nagoya University. So just first slide, I just showing the US omics-based clinical trial market, because omics is very important to development some pharmaceutical drug. In 2022, $19.5 billion is a good market for the global omics-based clinical trial market. And then also is very increasing step by step, maybe prediction the expanding the market is 16.8% each year. So now is mostly the pandemic for COVID-19 also is very useful for using omics-based clinical basic trial test. So what it means the omics-based clinical test? Here it's just showing first introduction is omics-based pharmaceutical analysis. Just a moment, please. So most of case, so natural product is a country. It's like Indonesia has maybe kind of the natural product. Maybe we put parallel source of the pharmaceutical activity lead compound, and also because of that their unparalleled the structure and unique biological activity. So omics-based technique. So here it's just showing some introduction of the omics-based the technique like genomics transfection or metabolites or recently very important bioinformatics method by using the supercomputer or chemical protect mixing or signaling or using some multi-omics based or label-free pharmaceutical protect mix method. So many kind of the mix method is very useful to finding some natural product for drug screening. But today just the point of the omics-based analysis, the multi-omics technologies are effective the tool of discovery the natural product target. And also chemical proteomics utilize activity molecular probe integrated some the MOA and biophysical method identify target of the natural products by changing of stability. So single cell techniques facilitate more piece of the real-time target identification. So this is the point of the omics based on the pharmaceutical analysis. In my case, I just focusing to water fluidity, such as aquaporin inhibitor is very important. Some reduce some product, reduce the disease for such as a neural some disorder. So next slide, just the introduction of the water in the vivo. In our life, the water is very important. So almost the 70% is the water is content in the human body. Also, the earth also 70% is the water. But this water is not so diffused. This is a little bit controlling by using some aquaporin. Here there's the how to finding aquaporin. That's the water the regulation on the cell. 80 years, mostly some science said this is the schematic of the blood red cell. This is a little bit different type of the cell compared to the other cell because it is the very highest permeability with water compared to other cell. Normal cell, just 0.0181 some water permeability coefficient. However, the red blood cell is very high permeability to the water. So the red cell is to put on the hypertonic solution. The water is just through out and then destroy the red cell. Then, or some put on the hypotonic solution, the water is just into the cell and then more expanding the cell. So it's the here is Professor Peter Agri. He originally wanted the eluted disease-related blood cell. In 1988, he gets one gene from the red cell, the name of the CHIPS28. This gene injected to the genopathy oocyte, and then he tried to changing the solution to from isonic solution to the hypotonic solution. In this case, normal cell not changing some cell condition, but after expressing the CHIPS28, this one changing the name of the aquaporin. And this aquaporin is very high water fluidity and easy to expanding. So he found in some CHIPS28, it's from the blood cell. This is one of the aquaporin. It's regulated the water fluidity on the cell. So what means the aquaporin? mostly is this is a schematic the cell membrane, and the water is just simply diffusion. It's very slowly in the inside outside water fluidity, but water channel aquaporin is a high fluidity and transmitted by the osmotic gradient. So and also here is just showing. Wait a moment. Oh, cannot. Okay. This is simulation with the water fluidity and the cell membrane. It's just so 20 million water is bisect. It's changing the inside and outside. Okay. So previous just showing it here and here. The Professor Peter Agre, he gets the Nobel Prize Chemistry 2003 to finding the aquaporin. And then the aquaporin, just mainly the two type. One of only pass the water, that just 0.6 to 54128. This is the normal aquaporin. The other group just not only aquaporin water, also some small solution like such as a glycerol or something like, and throughout the aquaglyceroporin type. So and then so many place the aquaporin is just expression and regulated some water fluidity. Also some aquaporin, some related some disease. Here is just one of the point of the aquaporin. The regulation is very important to reducing some disease. First figure is just showing, this is line, the survival rate, and this is time force. And this is wild type, aquaporin 4 expressions in the mouse cell, and this is knockout from knockout mouse. So effect of the toxicity in the survival is the AQP4 is very easy sensitivity and water fluidity, and then this cannot survival. But however, AQP4 knockout mice could be survival compared to AQP4 expression wild types the mouse. Why? So here the histological difference. Analysis is AQP4 expression, the mouse, the brain cell, or AQP4 knockout mouse cell after ischemia, diffusion. So this is the staining of some crystal violet brain, how to some destroy the brain function, and then the under panel just say the hemispheres and glutamate percentage. So it means some AQP4 expression cell is some how to say destroys part is very higher compared to AQP4 knockout mice. So maybe it's very important inhibit to the some water fluidity is to reducing some improve the survival rate. So very important AQP4 regulation by using some chemical compound or some natural compound. So how to finding the inhibitor of AQP4? So mostly people using some like this kind of the omics-based pharmacokinetic analysis. However, here today I just showing the two kind of the method for screening of the AQP4 inhibitor compound, because there is very high cost, also high. We need some high equipment or something like. So, but I developed a very simple method for screening method. Here schematic for the material and method. Here I just have to say we had prepared two kind of the cell line. one of AQP4 and joining the EGF. EGF like some marker of the fluorescence. And also we can try to make some combination in endothelin and cell receptor combination in AQP4. These kind of the gene expressed to the AQ in the CHO cell, and then after the expression of the cell to screening by using some freezing method. Mostly case of the cell that's by using the slow freezing by use it combined with some cell storage some solution. But in here it's point of the high ultra quick freezing. Normally most of the cell is the die because water makes some ice, and the ice is caused by cell apoptosis. However, in my finding, I have get one of the finding, and the blue line is the normal cell, and the red line is the AQP4 expression cell. This line is just survival rate percentage, and this line is the speed of the freezing. This is left side is slow, and the right side is very high. So mostly cell cannot survive on the liquid nitrogen. However, I finding AQP4 expression cell could be survivable in the liquid nitrogen. Here is just cell surface of the scanning electron SEM imaging. Here is non-freezing, and right side is quick freezing on the liquid nitrogen. And upper panel just showing is the normal cell. Here is the survival cell. And then this one could not survive, like just flattening the cell. However, AQP4 expression cell could be survival is both case in non-freezing or on the liquid nitrogen. So this one can be used in the screening test for some AQP4 expression cell. The blue one is a normal cell, and red marker is just a AQP4 expression cell. So I preparing some different rate of the AQP4 expression cell, and then after ultra quick freezing on the liquid nitrogen, and then again to sowing and culturing a few days, I finding only some AQP4 expression cell to survival after quick freezing. So we can screening the AQP4 cell by using on the quick freezing. Here it just also can be function the AQP4 expression cell. Just one. So left one is just showing the flow cytometry analysis quantities showing the population AQP4 positive cell. Here, this side AQP positive cell. Here is cannot finding the non-AQP4 positive cell. So and then here is the third time quick freezing or thawing, and then under panel is just non-freezing cell. So with the plasmid endothelin receptor combined AQP4 can be survival and also function of the calcium signaling imaging. However, non-freezing cell could not finding some endothelin receptor with AQP4 combination cell. So it's very quick freezing, also very useful to selecting cell for AQP4 cell. So just ordinary method, just we must screening some cell using label antibody, and then check the black one, and also culturing on the drug with drug and the drug screening, and then get some cell. But this method only just quick freezing, we can get some selecting the cell. So it's very low cost and reducing time for screening of cell. So maybe near future we can try to using how to finding some AQP4, how to finding the inhibitor of AQP4 is very useful for using this method. Some with some chemical compound for some, how to say, some inhibitor of AQP4 put on here, and then if the survival the cell, that it means some inhibitor is not function, but inhibitor is a function, could not be survival. So it's very easy to finding, and chemical compound can have the function of inhibitor of the AQP4. So, and then that one is the first result of the very simple screening method for using the quick freezing. So another one, I collaborated with Professor Chenkova providing a new method for biomarker for diagnosis. This is schematic of the cell, and then right side, but mostly the cell, not only some nuclear or something like, but mostly the occupy the water in the cell. So Professor Chenkova, she suggested aquapotomics method. Aqua means water, the poto means light, and omics like all. So water has so many all of information in the cell. So maybe water molecule mirror to be using as biomarker for diagnosis. She established this method is 2009. So mostly the scientists just focusing the solute, but so many kind of the solute or volume, but the solvent is only the water. So very useful to get information is from the, because water mirrors all of information with the solute. However, how to finding to the different water condition. I just today introduced solute solvent interaction behavior can be using near infrared spectroscopy useful to detecting the water molecule vibration and the clustering. So maybe near infrared spectroscopy very useful for development digital biomarker. So what means the near infrared spectroscopy? This one is very near the visual light, and then like LED light. And then this near infrared could be detecting some vibration of OH or CH or something like. Maybe this one is more easy understanding. Here is the schematic of the water vibration or clustering. So near infrared, some region could already assignment of V3 or V1, V3 combination, or some free water or something like. Some V1 means some symmetric stretching, and V2 is bending like this one, and V3 is just asymmetric stretch. So maybe we can try to pattern of the vibration or clustering water that depend on some disease or something like. Maybe we can predicting the disease or healthy by using some water vibration or clustering. So how to, but okay, just more introduction, the technical point of using near-infrared spectroscopy. Spectra is very simple, just only one line. So this method, the benefit is non-destructive, is non-damage of the cell, and non-staining. So we don't need some fluorescence or something like. And then very simple and continuous. So the near-infrared spectroscopy is has non-destroyed, non-staining, and simple method. But mostly this is absorption of the water vibration. So it's very difficult the assignment how to different water clustering or vibration. However, we had combination of some mathematical method to exclude some essence. So only just near-infrared spectroscopy spectra is hard to get some information, but combination is some PCA or semiconductor. This is some classification method by classification method or discrimination of some difference by using PLCDA, or we can predict some concentration or something like using some regression vector by using PLS method, or some we can grouping each or some similarity or unsimilarity by using cluster, just make some clustering by using some historical clustering analysis method. So mainly I using principal component analysis PCA by classification is some like healthy or unhealthy or something like. So this is what means the principal component analysis. PCA just is a spectra. So many kind of information. So we must reducing them information. So PCA method is providing transformation highly correlating the X variant like this one into the small set of uncorrelated related values. And also we try to reducing some noise, like this one may be noise or something like. And here is a similar some spectra. So PCA providing separated the derivative information from the noise. It means outlier. And then also here is just only just spectra, but the spectra can be how to say spectra changing some simple data like sample name or some each absorbance is each spectra. So so the raw data of the spectra can be visualized some score or loading. Score, it meaning like grouping in the difference. Like, for example, the factor 1 indicate is some gray group or black one, black circle. The factor 1 can be discrimination of the different color of the circle. And then what meaning of how to discrimination of the gray group or black circle. The factor 1 loading plot indicate which wave range important. Maybe this one or this one can be discrimination of the different color. So only just the spectra cannot say something right, but we can try to combination is some multivariate analysis. In here, just principal component method can be discrimination in the difference. So like with first time I tried to some different kind of mineral water by using this method. Each water try to detect some spectra, and then depend on the different kind of the temperature. So this one is the score plot indicate the factor 1 indicate a different kind of the temperature, and factor 3 indicate of the different color of the water. It means a different kind of the various water. So how to the factor 3 loading plot indicates some peak, gets some peak. Maybe these kind of peak could be discrimination of various water. What meaning of each peak? Each peak indicates some V3 or V1, V3 combination, or free water, or for water is a combined binding, like some ice. So this PCA method combined near infrared spectra combined PCA indicated some discrimination, indicated the discrimination of the various water from these kind of water vibration or clustering, like this one. So one of the publicated here. If you're interested in more detail, please reading this the paper. So spectra can be useful for discrimination in the different the various water. Now I try to using some the detect of some aging or some disease. So now it's just focusing the oxidative stress to screening some drug screening, because oxidase is cause some lipid oxidation or degradation or misduplication of the DNA, cause the cancer or some stroke or something like. And then this one to effect of the aging. The one here, this slide just mechanic of the loss reactive oxygen species. Oxygen some reacted to H2O2 and changed it to water. The other side, now we just focusing glutathione. Glutathione has the two kind of type. One of the reducing the glutathione type, we say the GSH, or this SH is the G-sulfido combined changing some oxidase glutathione, which called say GSSG. So GSH and GSH is important antioxidase function or some detoxing, such as some disease or something like, by using some glutathione conjugate function. So here is just introduction, and the glutathione GSH or GSHG is essential molecular redox balance in the related some disease. The first slide just showing the brain, the NMR. NMR can be detecting GSH concentration. This GSH concentration also very related some score. It's some neural disorder. Also some Alzheimer's disease, also very related some brain glutathione concentration. Is here is the right brain, and this is healthy, and then blue and red one is some Alzheimer's disease condition. So glutathione concentration is very related to some important Alzheimer's disease condition. Also, glutathione regulated some water fluidity in the by using some open closed stroma, and then redox very related some circadian clock in the plant. So glutathione is very useful for to finding some related some disease or water fluidity. So now it tried to applying the spectra to using to healthy or unhealthy cell to detect some glutathione concentration. Like left one is maybe some more healthy condition pattern, and the right side some put on some molecule, and then such as some disease, some how to say origin of the disease, and then water is a clustering and changing spectral pattern. So such as GSH more free GSH has more free water, and then GSH it means the oxidase the glutathione that like has some clustering water. So maybe the type of the glutathione reducing or oxidase is also changing to the water condition, the free water or clustering. Now we don't know why it's so different. Now it just try to detect, try to differentiate by using some MD simulation method. So near future, we can try to more reducing some oxidase stress, put on some chemical compound from nature resource. And then, so here it just must pharmacological method. So maybe Indonesia in the Lombok is very famous, some seaweed culture system. So seaweed culture market also very increasing compared than 2020. Built 16.7 billion US dollars, and then increasing some market in the seaweed cultivation. So what kind of the seaweed is very important? Recently, soybean is very popular for replace some beef or something like, and then, but soybean also seaweed, here is just focus on spirulina, a kind of seaweed, very high oxidant the function of soybean and seaweed, also, but total protein, the soybean also high content, but seaweed more high content or the comparison soybean. Also, the more easy growing the seaweed on the smallest area, also that one can be growing using the seawater. So very seaweed has very good function of the antioxidants, also high protein, also very more easy to cropping. So here I just indicate some spirulina has antioxidase activity on the mitochondrial activity. This is the intensity, and then here is the control, and then spirulina high antioxidase activity compared to control some other seaweed. Also, collagen is more productivity is the spirulina compared to control seaweed. So maybe spirulina has very useful for antioxidase some medicine or some more skincare of the collagen. Skincare of by using some spirulina material. And also spirulina could be protect antivirus. Virus is very like such a COVID-19 is very binding the receptor, and then to caused by some heat or something like. But the spirulina very protectant of the spike of the virus, and then the virus cannot do accessing the binding to the cell, and then so they can healthy. They cannot do the disease. So maybe this kind of the material to apply, such as some lipstick, is more very useful for how to say useful product compared to medicine. This is the schematic of the spirulina structure. This is the structure very important to maybe protect some virus. Also, spirulina can be useful for some colors, to some blue color. So in Japan, is very popular. This is one of the very popular. The ice cream is gari-gari-kun. Is very. I am also like this one. So if you visit in Japan, try to this one. Maybe this one, including the spirulina from seaweed. So just almost to finish the. So in future I want to make some aqua platform. So the like healthy or disease condition, how to different the water vibration or clustering. Also combined some other information by combined by other information. Also, we can try to some SDG problems such as the safety water or not can be tried to detecting by using near infrared. So visualization or patterning of the water molecule. So making aqua platform contributed extended by healthy longevity or some solving the SDG issue by using near infrared spectroscopy. So terima kasih. Okay.
Thank you.
Just a little bit high
Okay.
speed talking. Yeah.
Okay. Thank you, Kato Sensei. We go into the discussion.
Yes,
Maybe,
of course. Yeah.
Maybe anyone from the audience wants to ask questions.
If you had to talking, maybe to try writing chat or something like it's.
Yeah.
Okay. Yeah. Both is.
Yeah, you can write on the chat if you want. Even in Indonesia, it's okay. I'll translate.
Translate Japanese or something, right?
ちょっと難しい。
難しい? Okay.
Yes.
No problem. Okay. Please, Eka-san.
Eka-san.
お願いします。
Okay. Eka-san. どうぞ、Eka.
The mic.
Thank you very much, Kato Sensei.
Yeah, thank
so,
you.
I would like to ask my question first. Because I'm afraid I will forget, so I want to go first. So, it's a very interesting method in diagnosing a disease with the solvent instead of the solute, which we usually do, because if we want to diagnose a disease, usually the lab or physician, they take the blood sample instead of the water. So have you compared the results between blood sample analysis and using water analysis? How confident are you relying to based on water analysis to define a person's disease?
Thank you. It's just that we had two types of the method. One of the transmitted method, or some using the diffracting the spectra, and then transmitted spectra more good information, but the volume only 200 microliter. So very small hues of the water can be useful some get information. However, but we got some information from the 200 microliter blood cell or something like, but first time we can make some model by using, we need some reference data by using some biological method, such as we want to predict some sugar content in the blood. We try, first time we must try to detect some spectra. Also, we can try to detect some blood sugar content by using another method. And then we can make some mixing model But
yeah.
after
How
making
about
some maybe we can try to prediction in the near future, only just spectra. But
okay.
first stage, we need some more information by using, using some reference data. But the volume is very small, it's okay.
Yeah. You also compared the healthy cell and the sick cell from the water from healthy cell.
Yes,
That water which is that water from in vivo human, or is it from a cell culture?
Before I tried from the mouse blood cell.
Oh, okay.
Once
Mouse cell.
Mouse cell, and then try to the how to say that by using a needle, the mark to on it, and then try to directly to here, directly to the cuvette.
Oh, the human buccal cell, this part, the inner side of the cheek cell. Is it correct?
Just to compare some, in that case, some diabetic mouse or healthy mouse.
Oh, okay. Oh, diabetic mouse and healthy. Oh, okay.
Yeah. In that case, maybe like such as some water content a little different, also some sugar content also different. Glucose level is very different. And then we can try to more good information how to discrimination with the healthy or not. But in the near future, I want to try prediction of the diabetes. Yeah.
It's very interesting. Thank you very much, Kato Sensei.
Yeah, of course. Thank you. Maybe we can try to take some seaweed. I am not sure I understand, but try to take some seaweed, maybe more reducing some like reduce some diabetes or something like. We can try to comparing by using some spectra. Okay. Get some chat. So just wait.
Okay. Eka-san, do you want to ask any more questions?
I want to give the chance to the students first.
Okay.
Then
maybe the
I'll ask again
students.
to Kato Sensei.
Yeah. Okay.
Maybe to the students. Do you have any questions to Kato Sensei? Okay, we have a lot of students from various universities in Indonesia. So they're joining.
Yeah, Indonesia is okay. Maybe I get some translated.
Yeah. Okay. Does anyone have any questions?
I'm also interested in what kind of seaweed in the famous in the Lombok.
Yeah, it's called the brown seaweeds. I think there's a lot of brown seaweeds here.
Brown seaweed.
Okay. I have a question, Kato Sensei, if
Yes,
that's okay. Maybe while we are waiting. So I do a lot of research on the skin, and once I was reading the literature about wound healing, I think aquaporin is related to wound healing. If I'm not mistaken.
Yes, aquaporin very important to the recovery in the wound healing. Some in the skin, mainly AQP3 is very important. So maybe the edge area, like maybe
when it's proliferation.
Here is very need some rolling the cell. In that case, the water channel aquaporin 3 is very regulation, the rolling or some healing. So, but some of case AQP3 is not so good for healing because AQP3 regulates H2O2. So H2O2 also some of case is reducing an antivirus. But the first step is very important because after wound healing, some virus is some effect or something like. But the AQP3 the protect some antivirus. But after the healing, some of case not so good. because H2O2 is more effect, how to say, cause some apoptosis, something like. Basically hard to regulation is IQP3.
Oh, it's very hard. So is aquaporin on all the layers of the skin, or is it only on the epidermis?
Not only epidermis, and also AQP3 also, yes, I focus in the some other area also need some spread, like some glycerol transporter, something like okay, just one here.
Okay.
This one. Can I see?
Yes.
AQP3 also here. So many press the AQP3 also expression.
But it has the same function in anywhere.
Mostly the same function, yeah. But the expression level are a little bit different. Mainly the AQP4, AQP3 expressed on the epidermal cell on the skin. And also AQP4 is mainly the brain and the kidney, and the AQP0 only the eye.
Only
so
the eye.
Some aquaporin expression is very specificity of the body.
Okay.
Also,
so there's a lot in the skin.
A lot of skin and some mouth area, because more need some wetting or something like.
Oh yeah.
Yeah.
Thank you.
Yeah.
So there's a question here in the Zoom chat. It's from Fariza Izazi. I think she's a lecturer in Surabaya. So she was asking, I want to ask you about step to convert your data from NIR to PCA. Which data that you put into the PCA? Thank you.
Please, one more again, please reading.
So she was asking, what data do you put into the PCA for the principal component analysis?
What kind of data?
What data?
First time I just joined the mineral water, and the second time we can try to some in the case the glutathione is some using the chemical compound. Because we have some make a model by using some reducing oxyglutathione or oxidase glutathione component, chemical compound mixing in the water, and then try to detect some the difference by using the PCA method.
Okay, maybe to Mrs. Fariza. Okay. There's someone raising hand here. It's Muhammad Wisam Wira Sakti. Thanks from Brawijaya. Okay, this is a student.
Okay. Thank you from moderator. Good afternoon, Prof. Yasuhiro Kato. Thank you for the presentation. I have some question.
Yes.
Can aquaporin or AQPS as a biomarker or therapeutic potential to cancer model? Thank you.
Yeah. Cancer also very some effect of some promoting, because AQP4 expression is more promoting some proliferation. So maybe we get some inhibitor of the AQP4 or something like. Maybe also, I'm not sure, but cancer also effect of some proliferation. So maybe very important to some cancer regulation by using aquaporin. So near future, we won't try to prohibit the AQP function. It's more easy, more related to some cancer cell or something like. It's a good answer for you.
Yes, Professor. Thank you very much for the answer.
Thank you. We have several questions here in the chat Zoom. So this is good. From Siti Zainia. Siti Zainia from Universitas Mataram. Good afternoon, Prof. Kato Sensei. Thank you for the great explanation. She would like to ask about the method for biomarker for diagnosis. So there's aquaporin method, which is water. My question is how water can be used as a biomarker for diagnosis and the mechanism of it.
Biomarker diagnosis. My question: how water can be biomarker for how mechanism of aquaporin method? We have so many biomarkers. So now it's just the water is all including all information of the biomarker, but still it's very hard to establish method by only some water vibration. So maybe now we try to the step by step. Some previous just said that we can compare in some diabete or non-diabete mouse, some blood cell, how to different or not. So is that maybe, maybe one of the idea, some try to discrimination of diabete or not. So maybe if possible, try to the reading some paper more detail. The spectra can be assignment. Each wavelength, the different the water vibration or clustering. So we must try to get some more information, like some aging or not healthy young cell or some healthy the aging cell, maybe also different the water perturbation. So maybe now it's just step by step we get some information, the different the pattern of the water vibration. So so we have so many possibilities. Now we try to step by step to make some database by different kind of the situation. Is okay?
Siti Zainia, do you want to ask any more questions? Okay, we go on. There's another question from Nirwana. She's also a student. She's asking about, she read an article that the more aquaporins makes so more hydration, it makes water in and out of the cell. Does this mean that we can increase the number of aquaporins with a kind of process or something? Thank you.
Yeah, yes, aquaporin expression is the, I'm not sure the number of the aquaporin is the very hard to defining on the cell, but yes, maybe some increasing aquaporin expression, the number of the aquaporin in the cell increasing in the cell membrane, but I'm not sure how much expression or how much the number. I'm not sure that the number of the expression of the aquaporin. Maybe many kind, but I'm not sure. I'm not said the correct number.
Okay. Thank you. I think Mrs. Pariza was asking if she's can contact you by email to discuss her research, if that's okay.
Okay, yeah.
And thank you for the great explanation.
I just writing. If you need some contact, maybe try it here. Yeah, just send it. Yeah. Anytime it's to send some question. Oh, okay.
Okay. Someone raise hand.
Well, wonderful. Yeah. Please release the mute.
Okay, thank you for the opportunity, Mrs. Anggit. And I have question. What is an example of an inhibitor in aquaporin and how to fix it?
What kind of sample?
What is an example of an inhibitor in aquaporin and how to fix it?
How to disease?
To fix it.
Sorry, it's hard to. I need some, please, some help me.
Okay. Wanda, can you repeat your question? Or maybe you can type it.
Just some.
Sorry, can I use Indonesian language?
Oh yeah, of course.
Oke, terima kasih. Jadi pertanyaan saya itu apa contoh inhibitor yang ada di aquaporin dan bagaimana di aquaporin itu mengatasi inhibitor tersebut? Terima kasih.
Okay, so Kato Sensei, thank you, Wanda, for the question. Her question is, what are the examples of aquaporin inhibitors and what is the mechanism of the inhibitors?
Yeah, it's a little bit hard to explaining how to inhibit the aquaporin. One of the molecule is the mercury. Mercury is the prohibit the function of the AQP4. So maybe like mercury molecule, some structure or something like the effect of the function of the AQP aquaporin. But we don't know why the mercury is inhibit aquaporin function. That mechanism is still we don't know. But only one we know only mercury is some prohibit the water fluidity. Maybe just combine the poor position, but I'm not detailed. You are is okay.
You're happy? Okay.
Yeah. Thank you so much. That nice question.
Thank you for the explanation.
Maybe we want to try to finding a new inhibitor.
Yeah.
Yeah, from the
maybe
chemical.
from plants.
Plant, yes, the plant. Still, we don't know. So, yeah, we want to try using some seaweed or something like.
Yeah, interesting.
Yeah, please join in. Thank you.
Maybe other students want to ask more questions to Kanto Sensei.
Maybe, yeah, okay. Maybe if you need more information, question, please try to contact my email address.
In Keio.
Yeah.
Okay, maybe Eka-san, do you want to ask any more questions?
No, I'm okay. Later we can talk for Kata Sensei. I think for now for students, if you have any questions, it's okay. If you want to ask in Bahasa, you can use in Bahasa Indonesia and Dr. Anggit will translate for you. Nanti silakan bertanya dalam bahasa Indonesia, nanti dibantu terjemahkan sama Bang Anggit. Soalnya mumpung ada Prof. Kato di sini, jadi silakan digunakan kesempatan untuk bertanya. Tidak terkait mata kuliah aja, bisa juga terkait hal lainnya. Silakan.
Oh,
there's a question. Okay, so it's from Sumbawa. So it's from Sumbawa University. He would like to ask about omics in the detection and identification of biosynthetic pathways related to mycotoxin synthesis, because he heard a glimpse when his lecturer explained. Can you explain the basic mechanism?
Oh, toxin. Just a little hard for me to this question. Yeah, maybe later I can try to. Sorry, I'm not sure the detail of the mechanism of this method. Only I know the overview, so it's hard to. I'm sorry, I'm hard to the explaining of the basic mechanism of the mytoxics.
Okay. Okay.
I'm sorry. Yeah.
Andi Saputra, do you want to add any more questions? Is he here? Maybe there's more other students. Okay. Thanks. He says thank you.
Sorry. Maybe after lecture, maybe we need some talk with Eka-san. Yeah.
Maybe they are a bit shy. That's why.
Yeah.
Students.
In Japan, also many people is very shy and hard to. Yeah. But
Yeah.
after joining, they're really more easy cooperation. Yeah.
Yeah.
Hopefully next time to go the Lombok.
Yeah, you should come here again.
And then some try to discussing or something like.
Okay, does anyone have any more questions? Okay.
Okay.
Okay. If not, maybe we go on to the next session, Kato Sensei. I think we have a certificate of appreciation for your time. I think the committee will show.
Okay.
You can wait for the committee to show the certificate, please.
Okay. You're showing the here?
Yeah. We'll wait for the committee. Okay. Yeah, I think it's loading. Is it still loading or is it just on my screen?
Not yet.
Oh, not yet. Okay. I thought it was
Okay.
just me.
I can look it. Yeah. Maybe using
You can
the
say.
application. Yeah. No problem.
Yeah. No problem. So okay.
Oh yeah. I can see it.
So
Thank you
on
so much.
behalf of the committee, we would like to say arigato gozaimasu, Kato Sensei.
Yeah. Terima kasih. Yeah.
Thank you for your time. Mata aishita. We meet again sometime, and thank you so much for your explanation about omics technology. I hope it would give more insight to the students, especially I think there's a lot of biology students, pharmacy students, and it could give a good description of about the field. To be very useful. Thank you so much for your time, Kato Sensei, and we hope to meet you again sometime.
Yeah, thank you. Thank you so much. Yeah. See you again.
See you again. Okay, bye.
Bye. Eko-san, we talk something like here?
Hi. Yeah, you can stay.
Yeah, I can stay here, yeah.
Okay.
I just moving another place, so maybe we can try to continue the discussion. Just a moment, please.
Yeah.
Oh, okay, sorry. Wait. Kayaknya ada sesi foto ya?
Iya, lagi pindah tempat.
Kato Sensei. Oh. Kato Sensei?
Yeah.
Can we take a
Kato
picture?
Sensei. Before we close the session, I think you can introduce probably some scholarship opportunities to Keio University.
Yeah.
Do you have any information?
Yeah, Keio, yeah. I'm sorry, I'm not sure the scholarship at the Keio University, but maybe we can try some our homepage. We have so many providing some information scholarship to entrance here. But the Keio University, one of the very famous private school university.
Yeah.
Especially the medical school is the top of the area. So, and also we have so many kind of the area, law or some economy and some culture and the pharmacology or something like, yeah. So maybe if you're interested in our university, please joining here. And if we get some information on the scholarship, I also providing here again. So sorry, I now just don't have some information. So
I forgot to tell this also.
Yeah, sorry. Yeah, near future I'll try to check it. Yeah. Okay.
Okay. Can we take a picture first, Kato-san?
Oh, okay. Oh, yeah.
Yeah.
Of course. Yeah.
Okay. Say 1, 2, 3. Okay. Second page.
Okay.
1, 2, 3. Okay, thank you. Thank you so much, Kato-sensei. Thank you so much for everyone who has joined. And there's a link.
Ada postesnya. Mohon admin diinfokan postesnya.
Oh, ada postes.
I'm just moving here.
Yeah, okay.
Just move. Let's go. Okay, just starting again the discussion for collaboration for near future. Oh, so many students also stay here.
No, Kato-sensei.
Okay.
Wait, coba.
Okay. No problem.
Selamat sore peserta kuliah MBKM Farmasi Bahari. Mohon untuk diinformasikan bahwa pengerjaan post-test untuk soal post-test akan dikirim ke grup masing-masing yang ada dan batas waktu pengerjaan sampai besok sore. Terima kasih. Dimohon untuk meninggalkan grup karena perkuliahan sudah selesai.
Meninggalkan Zoom ya?
Iya, meninggalkan Zoom.
Post-testnya bukan besok sore. Tengah jam aja post-test mah.
Oh iya, maaf.
Butuh
setengah jam
waktu
saja.
untuk
30 menit aja. Udah dikirim belum post-testnya? Gampang kok itu.
butuh waktu untuk berpikir.
Ya, kalau besok sore nggak post-test namanya nanti.